Hello students, today we will learn basics of enzyme kinetics. Majority of the biochemical reactions are carried out by the enzyme inside the cell. Enzymes speed up the rate of chemical reaction without being consumed in the process. Enzymes are selective in their action. The enzyme binds to the substrate and forms enzyme-substrate complex. This complex undergoes a transformation and releases the product and free enzyme. The free enzyme is then offered for another round of enzyme reaction. Factors affecting the rate of enzyme catalyzed reactions. First, let's try to understand the effect of substrate concentration on enzyme activity. As shown in the diagram, the rate of reaction increases initially as substrate concentration increases, and then it gradually levels off to attain a maximum value at a relatively higher substrate concentration. It gives hyperbola curve. The relationship between substrate, enzyme and product is given by E plus S, that is enzyme substrate complex, and that it results into the enzyme and product formation. You can see the rate constant K1, K2 and K3. Now let's try to understand the Michaelis-Menten equation under steady state approach. What is steady state assumption? The rate of formation and breakdown of enzyme substrate complex are essentially equal. That is, rate of enzyme substrate formation is equals to K1 enzyme concentration into substrate concentration. Rate of enzyme substrate disappearance is equals to K2ES plus K3ES. At steady state, rate of enzyme substrate formation is equals to the rate of enzyme substrate disappearance. So that's why we are getting the result, that is K1ES equals to K2ES plus K3ES. Then ultimately we are getting what? On mathematical derivation, processing of this reaction, at the end we are getting this equation 3, that is E into S upon ES equals to Km. You know that E is concentration of enzyme, S is the concentration of substrate, while ES is the concentration of enzyme substrate complex, where Km is equals to K2 plus K3 upon K1. The amount of total enzyme, that is ET, is equals to the free enzyme plus enzyme bound enzyme, that is enzyme substrate complex. So we can see that the free enzyme is equals to the total enzyme minus bound enzyme. Substituting the value of the free enzyme in equation 3, we will get the equation 4. On processing, ultimately we will get the equation 5, that is Et into S upon Km plus S is equals to Es. Since rate of reaction, that is V, is equals to K3 into enzyme substrate complex, so on substituting the value of Es from equation 5, we will get V equals to K3 Et into substrate concentration upon Km plus substrate concentration. When all the enzyme is bound to the substrate, then total enzyme is equals to the enzyme substrate complex concentration, and at that point V is equals to Vmax. So we can say that Vmax is equal to K3 into Et. So Vmax is substituted for K3 into Et, and we will get the Michaelis-Menten equation. That is V equals to Vmax into substrate concentration upon Km plus substrate concentration. This Km and Vmax are kinetic constants and are characteristics of any enzyme catalyzed reaction under same condition. This reaction is very important in quantitative analysis of the enzyme kinetics. What are the significance of Km and Vmax? Km is defined as the substrate concentration, that is S, which results in the half of the maximum reaction rate. Km indicates the affinity of the substrate towards the enzyme. Km is a substrate concentration. Unit of Km is moles per liter or micromoles per liter. If substrate concentration is more than the Km, then we will be close to Vmax. Km indicates about the minimum concentration of substrate required for reaction. Vmax depends on the structure and concentration of enzyme. Km is inversely proportional to the affinity of the enzyme for substrate. Km is independent of the concentration of enzyme, but it depends only on the structure of the enzyme. How to measure the Km and Vmax? First of all, we need to carry out enzyme assay at various concentration of substrate and measure the rate of velocity. Then plot a graph V versus S. Estimate Vmax, calculate Vmax by 2, that is half of Vmax, and observe the Km from the graph. Now let's try to understand about the double reciprocal plot. Since from the Michaelis-Menten equation and plot, it is very difficult to derive the accurate value of Vmax from this plot. Similarly, accurate value of Km will also difficult. If we transform hyperbola curve into the straight line, then accurate determination of this constant is possible. So reciprocal of the Michaelis-Menten equation gives this equation 8. The plot of 1 upon V versus 1 upon S gives straight line. This plot is called Lineweaver-Burk or double reciprocal plot. Intercept of y-axis is 1 upon Vmax, while the intercept of x-axis is minus 1 upon Km, and from that we can easily determine the value of Km and Vmax, which is more accurate than the Michaelis-Menten plot. Now let's try to understand about the effect of concentration of enzyme on enzyme activity. When reaction contains the insufficient enzyme, increasing the concentration of enzyme speed up the reaction. To study the effect of increasing the enzyme concentration, substrate must be in excess. It should be independent of the substrate concentration. That is a zero order reaction. A reaction in which the rate of reaction is constant and independent of the concentration of the reacting substance, that is independent of the substrate concentration. We will get this graph. Now let's try to understand the effect of pH on enzyme activity. Increasing the pH initially increase the activity up to certain level. Then after increasing the pH will decrease the activity. pH at which maximum activity is observed is known as optimum pH. The graph gives a bell-shaped curve. pH affects the structure of enzyme. At optimum pH, ionization of certain amino acid residue result into most active structure of the enzyme. Such studies also help to identify amino acid residue essential to enzymatic activity. Generally, enzymes from acidophiles, neutrophiles and alkaliphiles have optimum pH in the acidic, neutral and alkaline side respectively. In this diagram, you can see that optimum pH is around 10. Now let's try to understand the effect of temperature on enzyme activity. Increasing the temperature initially increase the activity up to certain level. Then after increasing the temperature will decrease the activity. Temperature at which maximum activity is observed is known as optimum temperature. The graph gives bell-shaped curve. Possible explanation for such behavior of enzyme. It may be due to the effect on the stability of enzyme, effect on the velocity of breakdown of the enzyme substrate complex. It affects affinity of enzyme substrate complex. Temperature affects the structure of enzyme. At high temperature, enzyme denatures and the rate of reaction decreases. Generally, enzymes from the psychrophiles, mesophiles and thermophiles have optimum temperature range in the 5 to 10 degrees Celsius, 20 to 37 degrees Celsius or 45 to 70 degrees Celsius respectively. Means enzymes of the psychrophiles will work optimally in the low pH, low temperature, while the enzymes from the thermophiles will work optimally at high temperature. Similarly, in the given diagram, you can say that the optimum temperature of the enzyme is 50 degrees Celsius. Now, let's try to understand another important aspect of the enzyme kinetics, that is enzyme inhibition. What are inhibitors? These are molecular agents which inhibit or decrease the rate of enzyme catalyzed reaction by obstructing with catalysis, which are known as inhibitory agents. Inhibitory agents are grouped into two classes: reversible inhibition and irreversible inhibition. Reversible inhibition also includes competitive inhibition, non-competitive inhibition and uncompetitive inhibition. Let's try to understand each one by one. Competitive inhibition. This group of inhibitors exhibits inhibitory effect by non-covalent association with the enzyme. These inhibitors are substrate analog, means it resembles in structure with substrate. These inhibitors compete with the substrate molecule for the active site of the enzyme. That's why they are known as a competitive inhibitor. When inhibitor combines to the enzyme, then it prevents substrate from the binding. Such reactions are reversible. When high concentration of substrate is available, it will displace the inhibitor. So competitive enzyme inhibition depend on the concentration of enzyme and their affinity towards the active sites. You can see in the diagram that in competitive inhibition, inhibitors are binding only with the enzyme. In competitive inhibition, Vmax remains same, but the Km changes. This you can very well understand from the Lineweaver-Burk plot that is shown in the diagram. Another inhibition, that is non-competitive inhibition. Non-competitive inhibitors bind with both enzyme and enzyme substrate complex. Such inhibitors binds site other than the active sites. That's why inhibitors are able to bind both to the enzyme and enzyme substrate complex. A non-competitive inhibitor modifies Vmax while Km remains same. This kind of inhibitors are observed only for those enzymes which have more than two types of substrate. You can see from this diagram that enzyme is binding with the inhibitor is binding with enzyme, as well as it is also binding with the enzyme substrate complex. From this double reciprocal plot, we can see that in such kind of inhibition, Km remains same, but the Vmax changes. The next aspect is uncompetitive inhibition. Uncompetitive inhibitor bind with enzyme substrate complex. Such inhibitor binds site other than the active. A non-competitive inhibitor modifies both Km and Vmax. This kind of inhibitors are observed only for those enzymes which have more than two types of substrate. From this diagram, you can see that these inhibitors are binding only with the enzyme substrate complex. Now, overall, let's try to understand how inhibitors affect the parameters of the Michaelis-Menten equation, that is Vmax and Km. In the competitive inhibition, there is no change in Vmax, but Km increases. While in uncompetitive inhibition, Vmax decreases and Km also decreases. Non-competitive inhibition, Km remains the same, there is no change, but the Vmax decreases. Next aspect and the last, that is irreversible inhibition. The irreversible inhibitors binds irreversibly to enzyme. Such inhibitors forms a covalent bond with the enzyme. They may destroy the functional group of enzyme. Irreversible inhibitors may bind at the active site or site other than the different site. Many natural toxins are irreversible enzyme inhibitors. Example are cyanides, penicillin. These are my references. Thank you very much.